Hi guys, so today we will be talking about recombinant DNA technology. So basically this chapter is asking us how do scientists they take a human gene and make bacteria produce a human protein. So let's look at the general picture. Imagine a diabetic patient. Their body cannot make enough insulin. So our question is, how can we make insulin without taking it from another human? So scientists thought, what if bacteria could make insulin? And our question becomes, how do we teach bacteria to make a human protein? And this is exactly what recombinant DNA technology is. So first, our first step, here we can indicate this is our first step, is to find the gene. So where is the instruction for the insulin? It's inside a human DNA, right? So like if we go back to central dogma, DNA contains recipes and one of those recipes is insulin gene. And that is called gene of interest. So it's like a gene that we want. Insulin is just an example, by the way. It could have been insulin, it could have been growth hormone, it could be anything. So then our second step is our vector. So what analogy we can equate it to? So imagine you have a letter. You cannot just throw it into city, right? You need a truck, and truck carries the letter. And the same thing happens here. The gene needs transportation. The transportation is called vector. So vector is our second step. It's basically DNA that carries another piece of DNA. So this is vector. DNA that carries another piece of DNA, and one of the most common vectors is plasmid. So what is plasmid? Imagine a bacteria, and as you know, in the bacteria, beside their main chromosome, many bacteria have tiny circular DNA. So I can try showing you here bacteria plasmid. So you see here we have a bacterial DNA and then we have a plasmid. So this tiny circle you see here, this is called plasmid. And like scientists love plasmids because they are easy to modify. And our third step would be that we will cut the DNA. So you see here we have the desired DNA piece, and we are going to cut the DNA here so that we have a place to place it, right? So we will cut. How do we put the gene into the plasmid? They both have to be cut, but not randomly. So they have to match. So scientists for this, they use restriction enzymes. Restriction enzymes are used for this step, and they're like molecular scissors. For example, we have this. This is one of the most common examples. E. coli we have, and it cuts DNA at specific sequence. So it's always at the same place. And now we have both. We have our gene of interest and plasmids, and they are cut at the same restriction enzyme. But why? Why do we need to cut them with the same restriction enzyme? Because the sticky ends, they have to form. We have to form sticky ends so that the ends, they match perfectly. They naturally want to pair. And now you see here, now gene is sitting inside the plasmid, but it is loosely attached. So how can we secure it? So we permanently glue them together through DNA ligase. So we will permanently glue them together through DNA ligase. It's a glue. So just remember, restriction enzymes, they're like scissors. And DNA ligase is like a glue. Okay, moving on. So now we have recombinant DNA. So it means that we have DNA made from two different sources, human DNA and bacterial plasmid, but they're in one molecule. Step 5, host cell, you see here, we put it into bacteria. So now as we have our recombinant plasmid, but it's still sitting in a test tube, we need bacteria. So bacteria, they take up the plasmid, and this process is called transformation. So the process of transformation is bacteria taking foreign DNA. So this is the process of transformation. And how do we know that it worked? So we have a problem. Imagine 100 bacteria. Only 10 of them actually receive the plasmid. So it doesn't work on every single bacteria. So how do we know which 10? So scientists, they have a method for this. They put another gene inside the plasmid. Yes, they put another gene inside the plasmid. And for example, one of the most common genes that we use in such case is ampicillin resistance gene, because they grow bacteria on plates containing ampicillin. So who survives? Only bacteria carrying the plasmid, they are going to survive ampicillin. And then so this is a very clever idea, right? To identify, they will just put ampicillin resistance and gene, and they will grow it, and only the ones with plasmid are going to grow. So then bacteria, they multiply. So for example, from one bacterium we have 2, 4, 8, and can go millions and more. So now every bacterium contains insulin gene, and bacteria makes insulin. So as we have in the central dogma, DNA to RNA and then to protein, the bacteria read as human insulin gene, and then it make insulin protein. So now scientists, they collect the insulin and they purify it, and then they sell it as medicine. And I want to introduce something to you. We have a thing called cDNA. So what is that? It's we can also call it as complementary DNA. So I want to talk more about it. So the problem is human genes, they contain exons and introns, right? Human genes, they contain exons and introns. Exons have got useful information, but introns, they are actually removed before protein production. And humans, they know how to remove introns, but bacteria, they don't. So if we give bacteria the actual human DNA, it will be containing introns as well. Though in the final result in our protein, we don't have introns, but bacteria is going to get confused because it doesn't know it. It's going to produce introns as well, and that's an issue. So what do we do? What is the solution? Instead of DNA, we will be using messenger RNA. So, because messenger RNA does not contain introns, they were already removed, right? And then after that, after we have introduced messenger RNA, we have a process called as reverse transcriptase. Reverse transcriptase. So what is that? It does actually something weird, because normally we have DNA transferred into RNA. But as you can understand from its name, in reverse transcriptase, it happens the reverse. RNA turns into DNA. And now we get complementary DNA. And complementary DNA has got no introns and it's perfect for bacteria. So how can we get complementary DNA? So we will take RNA and we will turn it into DNA, right? And then this is going to give us complementary DNA. And then we have the zinc. I want to introduce you to the zinc. We have PCR. So in case you don't know, it is polymerase chain reaction. So PCR is polymerase chain reaction. It is basically a photocopier. Like we have 1 copy, 2 copy, 4, 8 millions. And PCR has steps. First, denaturation. Heat DNA and 2 strands, they separate. So first we have denaturation, where we heat DNA and we separate their 2 strands. And then we have annealing. We cool DNA and primers, they attach. And then finally we have extension. DNA polymerase copies DNA and it repeats 30 times, millions of copies. So this is why we need PCR. So in case we do not have enough molecule and we need millions, but like in a faster way, so we will just use this method: denaturation, annealing, and extension. If we just repeat it 30 times, we are going to get millions of copies. So this is just again a shortcut. So PCR is a shortcut for producing more genes. And then I want to introduce something to you. Okay, before that I will show you PCR. So you see here, creating millions or billions copies from a single trace sample. So here you see we have denaturation, annealing, polymerase extension, and new template, right? So, or you can see here. So this is basically how it happens. We can get a lot of more genes. So you can see here, you see denaturation, again first cycle, second cycle, third cycle, and when we have reached 20 to 30 cycles, we already have millions of copies. Isn't that so cool? Okay, and then I want to show you something: agarose gel electrophoresis. So what is basically agarose gel electrophoresis? We are going to talk about it. So the question is about agarose gel electrophoresis. So the question is, how do scientists check DNA, right? So DNA is negatively charged. That's an interesting fact. DNA is negatively charged, and electricity is applied, and DNA moves toward positive electrode. DNA moves toward positive electrode. And then what happens is that the gel acts like a kitchen sieve, and small DNA runs quickly, but large DNA through this process, it moves slowly. So after the run, scientists can estimate the DNA size, because the quick one is going to run quickly, but large will just move slowly inside of it. And scientists, they can get to know what is the size of DNA. And then plasmid. So this is basically the purpose of gel electrophoresis. So we through gel electrophoresis, we check plasmid size, we check if DNA was cut, and we check if insert is present, and we compare DNA fragments. So it's got a lot of uses. Now coming back, so then we have plasmid forms. The plasmids can actually have a lot of different shapes. So let me see if I can get illustration here. Plasmid shapes. Okay, so we have supercoiled. It's tightly twisted and it runs fastest in the gel. Then we have open circular. Its one strand is wicked and it's like relaxed circle, and it moves slower. And lastly, we have linear. It's cut in both strands. It is it's like, in the middle. So since these shapes move differently in gel, same plasmid can show different bands. And lastly, I would like to tell you something. And I've explained you about plasmid forms, and now I want to tell you about different types of plasmids. So let's talk about types of plasmids. So we have F plasmid. So it just transfers the DNA. And then we have R plasmid. It has got antibiotic resistance. Then we have cold plasmid. It produces toxins. We have virulence plasmid. It makes bacteria more dangerous. And we have degradative plasmid. It helps digest unusual chemicals. So opening of F plasmid is basically fertility plasmid. It carries genes for conjugation. It allows bacteria to form pilus and transfer DNA. R plasmid opening is resistant plasmid. It carries antibiotic resistant gene, and for example we have RP4 plasmid. And then we have col plasmid produced colicins. They are proteins that kill other bacteria. It produces, like we can say, colicins, basically a proteins that kill other bacteria. Then we have degradative plasmid. I didn't write that one. We have degradative plasmid. It allows bacteria to metabolize unusual compounds. So what do they mean by unusual compounds? So examples can be toluene, salicylic acid, and so on. For example, we have tol plasmid, of agrobacterium. Okay, and then how does conjugation happen? How does one bacterium give DNA to another? So conjugation, I want to introduce you concept. Conjugation, it's basically a plasmid transfer from one bacterium to another. So let's say donor cell has plasmid. So we do it by using a pilus. So donor cell has a plasmid. Donor forms pilus. I want to show you this as well. Conjugation and plasmid transfer. Conjugation and plasmid transfer. Okay, let's look at this one. So the donor forms a pilus, okay, and pilus connects donor to recipient, and plasmid DNA transfers. You see, plasmid DNA here, it transfers, and recipient gains plasmid traits, and now they both successfully copied and transferred. So, for example, if plasmid carries antibiotic resistance, recipient also becomes resistant. And lastly, I want to introduce you what is endonuclease and exonuclease. So basically, what is their difference? Let's talk about them. So endonuclease cuts inside of DNA. You can see that endonuclease cuts inside of DNA, and exonuclease, they cut from the end. So just the memory, so endo cuts inside and exo cuts outside. So now just let's go over the complete summary of this chapter. So we have human DNA. We choose insulin gene, let's say. We cut it with restriction enzyme, and then we cut plasmid with the same restriction enzyme, and then we match the sticky ends. DNA ligase glues them. Then we form recombinant plasmid, and then we transform it into bacteria, and then we select using antibiotic resistance. Then bacteria multiply. Then they make insulin, and then scientists, they purify insulin and they use it in the medicine. So that's it for this video. I hope you liked it and I hope you understood. I'll see you in the next video.